Hi guys, welcome back. It's been a while. Didn't do videos much lately, but you may remember that the last time I was doing that, we were talking about vaccines for Corona. We're talking actually a lot about mRNA technologies. Quite some videos were uploaded back then. And now it seems like we're getting really close to those technologies to actually work and to mRNA vaccines to be launched onto the market. And the closer we get to that point, the more voices I hear, the more questions I get about the potential dangers of this new technology, about how it might affect our genome, about how we're manipulating our genes. And I think it probably is good for all of you if we have like one video where we discuss this a bit more in detail. Some parts of it are easy to understand, and then we have to go a bit more into detail, and it gets a bit messy and confusing maybe. So maybe a video is the best way to do that. Okay. So maybe let's start by you getting familiar with the keywords you need to understand during these couple of minutes of video. The easy ones are genome, DNA, RNA, and proteins. And if you are actually one of my students, then it's very likely that you have heard them plenty of times. What you might have heard a bit less are these guys here: retroviruses, reverse transcriptase, integrase, and primers. And they all play a role now when we try to figure out how dangerous mRNA vaccines actually could be or could not be. So first of all, let's discuss what actually has happened now in these last 6 months. Well, good stuff has happened. I introduced that technology to you. I told you that it sounds very promising, and it seems to be a great technology. I've discussed it a lot in class already. I'll quickly summarize. Basically, you've probably all have heard these headlines. We have Moderna, we have BioNTech, Pfizer, who published close to 95% efficacy rate in their trials, right? So to understand what that actually means, you need to kind of realize how these trials are performed. So let's say you're Pfizer BioNTech and you want to do a nice trial 3 testing. That basically means that you will recruit 40,000 participants. I think they had 44,000. You divide them in a placebo group and an actually test group. And so basically to the one group you'll give a vaccine, to the other group you'll give them something which looks like the vaccine but is actually not vaccine. But no one really knows which group is which, right? So the people don't know, the doctors don't know. That's what you call double blindfolded testing. Okay, and then all you do is to basically look how many people get sick, and when you reach a certain number of people getting sick, I think for BioNTech Pfizer it was 170 something, then you unfold, you look at it, and you see, okay, out of my 100, let's say it was 100 people, out of my 100, you have, in the case of a 95% efficacy, you have 100 people who got COVID-19, 95 of them will be in the placebo group. Okay, whereas only 5 were in the actual vaccine group. Okay, we're working with quite high numbers overall, and what this basically means is if you get the vaccine, it gives you a 95% lower chance to get the disease. So this is a good statement, is a strong statement, and is a lot higher than what has been expected before. Okay, and both BioNTech and Moderna pretty much have the same results here, already showing that the mRNA technology seems to be very, very efficient here. You might have also heard of AstraZeneca, but AstraZeneca is doing a completely different technology, and we will not discuss that now, right? Right now I try to focus on mRNA technology. Okay, now that we already know it seems to work very, very well, we need to understand how dangerous it is, right? That's the other thing. Now, they started the big trials in July. In these 4 months or whatever that is now, they didn't have any bigger side effects. Okay, so that's already a really strong statement as well, because we're talking about 45,000 people for BioNTech and another 30,000 for Moderna. So basically, it has been already tested on almost 100,000 people over 4 months, and nothing has been seen yet. Okay, but this alone obviously cannot exclude potential risks along the way, right? And the fact that we're talking about RNA and somehow manipulating your cell to do certain things obviously sounds scary, and we need to discuss the details of that. So, first of all, most of you know, some classes don't yet, need to understand what DNA, RNA, and proteins are. Okay, DNA, what I'm showing to you here is the dogma of biology. Okay, DNA is the very base of your cell. The DNA is the blueprint. The DNA is basically what gives your cell the information to look what it looks, and in the end gives the information to your entire body to look the way it looks, to work the way it does. But frankly, the DNA doesn't do very much except for replicating itself. And then the only other job DNA has is to create RNA. It's done with an enzyme called RNA polymerase. Okay, so the only job of the DNA right here is that it has certain genes, which are little sections on your DNA. So that, for example, would be a gene. On your chromosomes, you probably all know chromosomes. So you have little sections of little sections of DNA on your chromosomes, and these sections are called genes. And these genes get transcribed into RNA. Then this RNA wanders around your cell and is in turn again translated into proteins. And proteins, that's actually the real deal, because proteins make you. Whatever you look like, whatever you are, this is done by the proteins, right? So everything you see in this man's face, the color of the eyes, the form of the nose, the way the person smells, the way the person talks, all this is done by the proteins. Okay, so the DNA is the blueprint, but the proteins is the actual build, the actual function. Everything which happens, that's the proteins. And the in-between, that's the RNA. Now, what we do with those mRNA vaccines is to kind of hijack that system, okay, which is the point which scares people. So we basically have an RNA which codes for the viral protein. So in particular, it codes for the spike protein, which is the protein the virus needs to bind to your cells, but it doesn't matter. Basically, we produce a part of the virus in you. The idea behind that is that vaccines work like that. You need a part of the virus in you so that your immune system can be triggered against that virus. So it basically gets ready to fight that virus. Okay, so instead of introducing the entire virus, which are other vaccine technologies, we introduce just a little RNA and let your body synthesize the protein of that virus. Okay, and I'm not going to discuss the details of this again. I do that in a lot of detail in the other videos I uploaded half a year ago. But basically the end result is that little pieces of the protein are presented on your immune cells, and by that you trigger the immune system. So what people now are afraid about is you can actually subdivide in different levels depending on how much people know about biology. If you don't know much, then probably what you feel is that you're really scared about the fact that you put a piece of genetic material called RNA into your cell, right? So my cell, and you want my cells to produce a piece of virus, so basically completely genetically modifying my cell, right? It's like making me a GMO, right? Of course, that doesn't sound good, okay? So here's what we need to understand at that stage. This is not happening. Let's go back to the slide we saw before. And let's understand that what our genome is. So when we talk about our genes, that is the chromosomes, that's the DNA we have, okay? DNA strands.